What would happen if cells worked without any control? Cells will produce too many products, waste energy, and lose balance. This is why nature has a powerful control system called enzyme inhibition. In this video, we shall explore how enzymes work, the differences between competitive and non-competitive inhibitors, and how these inhibitors affect the rate of enzyme-controlled reactions. Okay, now let's get into it. Now, every second, thousands of chemical reactions are taking place inside living organisms, and enzymes are biological catalysts that make these reactions occur or happen even faster to support life. However, enzyme activity is not always allowed to continue freely. Cells have a way of controlling enzymes and how many products are formed, and this important method is what we call enzyme inhibition. So, enzyme inhibition occurs when a molecule called an inhibitor reduces or stops the activity of an enzyme by interfering with its ability to bind with the substrate. Now, this process is essential for regulating metabolic pathways, controlling the amount of products produced, and maintaining some sort of balance within our cells. Okay, now there are two main categories of inhibitors, and these are competitive inhibitors and non-competitive inhibitors. Now, under non-competitive inhibitors, there are two categories, and these are reversible inhibitors and irreversible inhibitors. Okay, now I'll start with competitive inhibitors. Now, these have a molecular shape that is complementary to that of the substrate, and this allows them to occupy the active site of an enzyme. So because they have the same shape as the substrates, these inhibitors can therefore compete with the substrate for the available active site, and this may prevent the enzyme from combining with the substrate, hence reducing the rate of reaction. Okay, now an important thing to note here is that the effect of a competitive inhibitor depends on its concentration. So the higher the concentration of these inhibitors, the more the more they will reduce the rate of reaction. So this means that increasing the substrate concentration will lead to an increase in the rate of reaction. This is because as you increase the substrate concentration, these will effectively compete with these competitive inhibitors for the active sites, for the available active sites of the enzyme, and therefore our rate of reaction will therefore increase. And that is what you essentially need to know about competitive inhibitors. Nothing to add there, unless of course you have something to add, but that's what you need to know. And next I'll talk about non-competitive inhibitors. Now these have a shape that is not complementary to the shape of the active site of the enzyme, and therefore, since they don't have the same shape as the substrate, they don't compete with the substrate for the active sites of the enzyme. Now instead, these non-competitive inhibitors bind with the enzyme at another site other than the active site, or at a site different from the active site, and this is what we call the allosteric site. So these non-competitive inhibitors bind with the enzyme at the allosteric site, and this causes a change in the shape of the active site in such a way that the substrate can no longer bind with the active site of the enzyme. Now from the illustrations I've put there, you can clearly see that after the non-competitive inhibitor binding with the enzyme, there's a change in the shape of the active site, and that alone prevents the substrate from combining with the active site of the enzyme. I know some of you are visual learners, so I hope that one brings whatever I'm talking about to life, or it helps shed more light what I'm talking about. So here, under non-competitive inhibition also, you need to know that here an increase in substrate concentration does not decrease the effect of the non-competitive inhibitor. So even if you increase the substrate concentration, it won't help to increase the rate of reaction. This is because the shape of the active site has already been changed, and therefore even if the substrate manages to combine with the active site, there will be no change in the rate of reaction. And even if it eventually binds with the active site, catalysis cannot occur. Now if you want to know what catalysis is basically the speeding up of a chemical reaction using a catalyst, which is our enzyme in this reaction. And that is we can now move on to reversible inhibitors. Now at the beginning of this video, I said non-competitive inhibitors are grouped into reversible and irreversible inhibitors, and I'll start with reversible inhibitors. Now reversible inhibitors, clearly the name gives them away. Okay, this whatever effect these have on an enzyme can clearly be reversed from. That's the basis of their name. Now reversible inhibitors, these cause a temporary change in the shape of the active site of an enzyme. So they change the shape of the active site of an enzyme in such a way that the substrate cannot bind with the active site of an enzyme. So even if the shape of the active site is reduced, eventually a substrate can bind with this enzyme, but because of the presence of the reversible inhibitor in this case, even if the substrate binds with the active site, no catalysis will occur. And I hope you know what catalysis means. Anyway, reversible inhibitors affect the enzyme and the rate of reaction only as long as they're attached to the enzyme. So as soon as these reversible inhibitors detach from the enzyme returns back to its original shape and can function normally. So that's what you need to know on reversible inhibitors. And finally, I'll talk about irreversible inhibitors. Now I'll be brief here. Now these cause a permanent change in the shape of the active site of an enzyme, and therefore these stop catalysis completely. So as long as this binds to an enzyme, the enzyme stops functioning there and then. And even if a substrate binds to the active site of the enzyme, despite the change in its shape, you realize that no catalysis will still occur. So this causes a permanent change in the shape of the active site of an enzyme. Now, most irreversible inhibitors are harmful, and these may include toxins and poisons. I'm saying most, but not all. And that brings us to the end of our journey into enzyme inhibition. Now, if you found this video helpful, make sure you like the video, subscribe, and join me in my next video for more fascinating biology explanations. Thank you for watching.